Neuropeptide Y is a neuropeptide. Neuropeptides are made up of amino acids. Neuropeptide Y is 36 amino acids in length and plays a crucial role in whole body energy regulation. NPY was first discovered from a pig's brain in 1982. The study of NPY in adipose tissue has been the focus of many studies recently. There are various factors that contribute to adipose tissue, but it was recently discovered that the expression of NPY in visceral adipose tissue was one of them, and that NPY stimulated the proliferation of primary rat pre-adipocytes. Collectively, the studies all suggest that in mammals, NPY systems promote energy storage in adipose tissue. This experiment demonstrated that direct injection of NPY in chickens would stimulate food intake. It was found that depending on the chicken's weight, the NPY had different effects. Because of this, the hypothesis was that NPY systems plays a role in lipid accumulation in the adipose tissue of chickens. The objective was to investigate the role of NPY in adipogenesis. A fast-growing boiler line was used as the model in this experiment. It was reported that neuropeptide Y stimulates the proliferation of primary rat pre-adipocytes and 3T3L1 mouse pre-adipocytes. Further studies into the 3T3L1 pre-adipocytes and sympathetic neuron-derived tumor cell co-cultures show that neuropeptide Y promotes pre-adipocyte proliferation and differentiation through neuropeptide Y receptor subtype 2. Neuropeptide Y promotes energy storage in adipose tissue through both central and peripheral pathways. It was also determined that neuropeptide Y may affect adipocytes through the neuroendocrine, endocrine, paracrine, and autocrine routes. Neuropeptide Y stimulates food intake in vertebrate species and promotes adipogenesis in mammals. This study was done to determine if neuropeptide Y promotes adipogenesis in birds as well. Originally, there was no effect of NPY on proliferation within the first 12 hours after the treatment. During the first 6 days post-induction differentiation, there were effects on proliferation and lipid accumulation due to the NPY treatment. It was found that there was a greater glycerol 3-phosphate dehydrogenase activity when there was a neuropeptide Y supplementation. This was discovered through the use of preadipocyte markers and discovering that there was a greater amount of proliferating cells in the groups that were treated with NPY. It was concluded that NPY is likely to play a role in the promotion of adipogenesis in chickens, which is done through the increase in synthesis of preadipocytes and lipid synthesis and storage. We are going to go through the methods and materials briefly. All protocols for animal treatment in labs were followed in this study. We began with 2 grams of abdominal fat, which was collected from 14-day-old male chicks. This tissue was then minced into fine sections and kept at 37 degrees Celsius in 10 milliliter incubation tubes. The tubes were centrifuged and separated in various ways. Cells were then seeded onto petri dishes. The petri dishes were then treated with either 0, 1, 10, or 100 nanometers of chicken NPY, which was synthetically created. These cells were then cultured and stained with oil red O, and then digital images were captured to determine the results.
G3-PDH activity increased as the amount of NPY present increased when day 6 was reached. An increase in NPY was accompanied by an increase in fatty acid binding protein 4 mRNA and an increased expression of lipoprotein lipase. All of these bar graphs show how NPY affected the folding of mRNA with different gene markers. You can see that while NPY increased the difference in the enhancer binding protein beta, FABP4, LDL, Chi67, TOP2A, TPX2, and GATA2, the remaining mRNA were either decreased or relatively unchanged after 6 days. This graph shows the presence of NPY as NPY treatment increased. The dramatic rise in mRNA fold difference suggests a positive feedback effect of NPY expression in these adipocytes. This figure is a result of staining chicken adipose cells with oil red O stain. It is clear to see that there was an increase in the number of adipose cells as NPY increased and days went on. This is another figure that shows two different fluorescent stains of chicken adipose tissue. Here it can be seen that in both stains, the amount of cells increases as NPY increases. As you may have already noticed, under each graph is a p-value which represents statistical significance. In this case, results are statistically significant if the p-value is less than 0.05. The red underlined values show that the results on these graphs are statistically significant enough to back up our claim. Overall, we can say that higher amounts of NPY generally produce more adipose cells as well as change the folding of mRNA that affects adipogenesis. The best results given were typically after 6 days had gone by. Chi67, TOP2A, TPX2, GATA2, FABP4, and LPL results all gave us important statistically significant results that support our hypothesis. Since this is the first study that shows how NPY promotes adipogenesis as well as general adipose cell function, our understanding of how these factors that affect NPY and adipogenesis in chickens was greatly improved as a result of our research. The importance of this study is significant because the amount of research on adipogenesis in chickens is very minimal compared to mammals. This study shows that adipogenesis can occur through NPY in chickens more similarly to mammals than what we may have thought before. However, it is still unclear exactly how gene expression affects the transcription factors that regulate adipogenesis in chickens, because we still don't know as much about chicken genes as we do mammal genes. Overall, as a result of this study, we now know NPY can promote adipogenesis in chickens, but we're still not 100% sure which exact transcription mechanisms are responsible for this, since they are slightly different than mammals. This research can be applied to increase our understanding of how neuropeptides work in chickens, as well as specifically how NPY works as an appetite inducer in chickens. We can use this information to compare and contrast the genes of chickens to what we already know about mammal genes. Now that we know more about NPY function in chickens, we could one day use chickens as a model for obesity, anorexia, or other eating disorders. Increasing our knowledge about eating disorders, even in a model like chickens that is not as similar to humans as a mammal model, could still help to figure out the biological cause of certain eating disorders, as well as even coming up with treatments. Additionally, knowing more about chicken appetite through NPY could possibly help come up with the most efficient way to feed chickens and reduce costs in the agricultural industry. Any reduce in cost, even in the slightest, in such a big industry like chicken production, could spark a major change in the economy and positively affect the entire agricultural industry. This study does well at showing the changes of many different factors that relate to adipogenesis as a result of an increase in NPY. However, it is possible that not enough time was given for mRNA folding of each factor, and providing more data over longer periods of time could have bolstered the credibility of results. Additionally, there could have been multiple confounding variables that caused an increase in adipose cells, including the type of environment the chickens were in, as well as other biological markers that were not tested. Both collecting data over a longer span of time, as well as reducing the amount of variables that could also affect adipogenesis, would greatly improve the significance of this study.